Good day to everyone. Thanks for the organizer for the invitation to share my thought on tropical plant improvement using precision biotechnology. In this talk, I will break it down to 5 parts. First, let's define precision biotechnology. Then we focus on plant improvement in tropical countries, especially Malaysia. Then I'll touch on some of the challenges and prospects of precision biotech. What is actually precision biotechnology? Perhaps the first thing that comes to your mind is genome editing. However, it also encompasses all the latest development in biotech, namely genome-wide association study, molecular breeding, functional genomics, high-throughput genotyping, and phenotyping, which are applicable across industrial, environmental, medical, and agriculture. In the context of agribiotech, this leads to an important aspect of precision agriculture apart from automation and mechanization through IoT or IR4. My talk will just focus on gene editing today. Genome editing tools have come a long way from TFO, zinc finger nuclease, tail nuclease to CRISPR technology with increasing specificity and feasibility. Genome editing tools have been applied for a wide spectrum of molecular manipulation from transient insertion to precise single-space editing. They have wide-ranging application in various aspects of biotechnology, from basic research, therapeutic gene editing, to bioproduction across all kingdoms. Keep to the theme of NCPBA, I will discuss only on plants. Just a brief recap on the basic principle of nuclease-based genome editing, which allows knockout and knock-in through gene targeting. It also generates genome instability resulting in indels of long DNA fragments and also chromosomal translocation. You can see here the knockout in either non-coding or protein coding fragments, and also knock-in by insertions. Okay. The CRISPR-Cas system was discovered in less than a decade with an unprecedented speed of development. There are two aspects of CRISPR biology. Firstly, the adaptive immunization from the previous bacteriophage invasions using different set of Cas. And then there is the adaptive immunity for the defense aspect that has been utilized for genome editing. So this is what we are used to on the genome editing. So CRISPR are the remnant palindromic sequences from previous viral invasion found interspersed in the bacterial genome. This animation will kind of briefly summarize what I said just now. This spacer provides defense against repeating bacteriophage infection. So during the invasion, the pre-existing remnant going to guide the CRISPR RNA to destroy the invading bacteriophage. And then for new infection, a short stretch of the sequence will be incorporated in the genome for future defense. Hence, this is known as the adaptive immunity for bacteria. CRISPR mechanism has been exploited to serve as a guide sequence for the amazing Cas9 nuclease to scan for complementary sequence in the whole genome for cutting as a molecular scissors. In such a way, it permits precise gene targeting and at the same time can also confer interference, known as CRISPR, by blocking gene transcription. A customizable library of single guide RNA can be built to target different genes simultaneously. The original Cas9 is discovered in Streptococcus pyogenes, and it since has been developed with various variants like the high fidelity mutant, Cas9 nicases, dead Cas9, and Cas9 fusion. The original, all of these are from the type 2 class 2 CRISPR system. The original Cas9 had the NGG pump PAM site and with 20 nucleotide protospacer and double-stranded cut with blunt end. In the beginning, it was with the tracer RNA and the CRISPR RNA, but now it's developed to just use a single guide RNA for functioning. Apart from that, some smaller and simpler RNA-guided DNA endonuclease CPF has been used recently, and they have different features. Class 2 Cas nuclease are diverse. You can see here. Cas9 and Cas12 target double-stranded DNA, and Cas13 target single-stranded RNA, so they contribute to genome and transcriptome editing. So Apart from the genome editing, which is on top here for the cleavage, there are also various other broad range of application in DNA and RNA targeting. DNA targeting and this RNA targeting, so the left and right. So this Cas effector can be turned into RNA-guided DNA or RNA binding domain by inactivating their catalytic residue, known as the dead Cas. So all these are from inactivated catalytic domain. So dead Cas can be fused to a variety of functional moiety to achieve targeted repressions, activations, epi modifications, base editing, and imaging at either the DNA or RNA levels. So it can use for splicing regulation as well at the RNA levels, also on the nuclear organization. So CRISPR toolbox has also widely applied to target different elements of the gene, either in the coding or the non-coding element, and especially in drug discovery. So we have various tools. This shows the timeline of arbrosis genome editing is now moving toward a cell-specific gene targeting. With the basics of CRISPR-Cas in mind, we move on to the plant improvement, which is essentially driven by genetic variations, either natural or induced. Nowadays, we have more targeted, fast and efficient genome editing for modern agribiotech crop improvement. There are many examples of desired crop improvement, including resistance against diseases and environmental factors, or crop with higher yields and better nutrition or without allergens. For traditional breeding, natural transgenic to man-made transgenics, the field has come a long way. Now plant molecular biologists can target gene editing responsible for certain desired traits. These are some of the examples. And this provides an overview of genome editing tools with the possible genetic outcomes and crop trait generated using these tools. So I'm not expect you to look at this. You can refer to the paper if you are interested. It's worth noting that genome editing can be stable or transient based on the different delivery approaches, and we'll come back to this discussion later. In general, the workflow of crop improvement through genome editing is very similar to that of transgenic technology, which differ mainly in the conformation or editing events. And this is where genomic analysis becomes important due to the possibility of off-target editing. So off-target analysis has become part and parcel of genome editing pipeline as illustrated here. And nowadays, off-target analysis on the Cas9 digestion site can be performed using long-read technology, such as PacBio and Oxford Nanopore technology, without the need for gene amplification. For off-target analysis, a precise reference genome is essential. Hence, that's why population resequencing is becoming important to capture the genetic diversity in different varieties of crop, which are not present in the reference genome. This allows genome editing to be targeted for different varieties. For example, the study on genetic architecture of selected traits in bitter gourd, published in June. When multiple genomes are available for a single species, a pan-genome, which includes all the genetic diversity, can be constructed in comparison to the core genome, which excludes the accessory and unique gene in certain individuals. A nice example of pan-genomic is the recent pan structural variants based on long-read sequencing of 100 accessions of tomato. Many structural variants in the cis regulatory element are identified to be responsible for the flavor, fruit size, and productivity of tomato, which otherwise won't be detected based on a single reference genome study. Another interesting take from the study is that often a mutation in the open reading frame will have a drastic qualitative effect, present or absent, whereas the mutation in the non-coding regulatory elements provide a range of quantitative variation to tweak on, such as the finding by Lippmann group on tomato size based on targeted promoter editing of the clavata3 genes. Now we look at a case study on rice with extensive publication of genome editing study. This case study is mainly based on a book chapter recently published by Mehta et al. This table explains why CRISPR-Cas is currently dominating rice improvement in nuclease-based genome editing, mainly because of the ease of use and higher success rates. Rice genome editing follows the conventional transgenic approach. We rely on the tissue culture of the calli and plantlet regenerations. We have many CRISPR-Cas tools and software developed for the design of single guide new RNA and off-target analysis in the table here. There are also CRISPR-Cas database for the curation and reference. And there are many commercial companies have joined the implementation of CRISPR-Cas. Worth mentioning here is the Origins, which supplies synthetic single guide RNA. Here are some of the reports on CRISPR-Cas mediated rice improvement for increasing yields, like larger grain size, dense particle, and also increasing stress resistance, for example, to bacterial blight and blast resistance. Also for nutrient improvement for enhanced amylose content as well as reduced amylose content, also to enhance fragrance. The improved variant of Cas9 CPF1 also been applied in rice, and this includes various functional studies with potential higher efficiency rate. And to conclude the rice case study, genome editing has proven success in varietal development for introducing desired trait in the existing elite rice germplasm, which will hopefully generate super rice to feed the demanding world populations. CRISPR-Cas genome editing has been evolving very fast with new technology emerging now and then. Here we look at the latest development in the field. Aside CRISPR-Cas9, other approaches using base editing and prime editing are more recent with better specificity, but they all still suffer on off-target genetic alteration, which require off-target analysis. So, for precise base editing, we have cytosine and adenine base editor for single base mutation. Why perform only single base mutations? This is because many important agronomic traits involve only one or a few base changes. And here you can see the NRT 1.1b control the nitrogen use efficiency, whereas the OGTT1 control the heat tolerance in rice. The more precise base editing is therefore suited for the precision molecular breeding for the single base mutations. Other examples include this in rice, also in Arabidopsis. The most recent game changer is the prime editor, which allows the introduction of all mutation types, including indels without double-stranded breaks or the need for donor DNA. The Cas9 nickase is fused with a reverse transcriptase and works along a prime editor guided RNA on target sequences. Prime editing opens up opportunity for various applications, including directed molecular evolution and protein engineering, apart from other existing genes manipulations. Multiplex genome editing is also becoming more common to target multiple genes simultaneously. As most plant genome editing still relies on in vitro transgenic technology, the delivery of CRISPR-Cas into the cell remains the biggest challenge today. Same applies for other CRISPR-Cas delivery systems in mammalian cells, also in microbes. There are many strategies, like in plant as shown here, to deliver the CRISPR-Cas for the genome editing. For details, you can refer to the recent review by Prof. Gao Caixia. And with regulatory concern on transgenes, the field is moving towards transgene-free editing approaches using ribonucleoprotein. You can see in this using the particle bombardment. There's also certain construct related for self-excisions. So this is example that this DNA cassette will be self-excised out in apple when induced by heat with minimum trace of transgenes without antibiotic marker. Another alternative is to utilize a pigment-based marker to replace antibiotic marker for the selection of transformant. For example, in this betaline synthesis, this is common in dragon fruit. If you notice the purple color of the dragon fruit, this is from the betaline, and it's actually simple pathway by the 3 genes we can produce the betaline compound conferring the purple pigment. There are also a list of other selection-free approaches used in genome editing, mostly based on protoplast transfection and particle bombardment. However, these suffer from low mutation efficiency. Recently, there's a new way to improve transformer regeneration with utilization of a growth regulating factor chimeric protein, and this is shown to be successful in wheat for the transformer to have higher regeneration. The field of CRISPR-Cas is still rapidly developing with innovations such as this compact cascade Cas3 system for large segment of DNA modification. This is shown in yeast, not yet applied in plant. So now come back to the precision biotechnology in the tropical context. How this can be applied in tropical countries, which are mainly developing countries like Malaysia. Tropical plants are diverse and many are lacking genomic information. Despite that Malaysian researchers are well capable of adopting genome editing in crop improvement, but initiating such a project won't be cheap and require long-term financial support. Whether the products are marketable will depend on acceptance of the society, and this link to the values and demand. Are the values and demand sufficient to justify for the investment in precision biotech? Just like the argument for investment in precision medicine. Furthermore, the regulations of genome editing is also an important determinant for the uptake of this technology here. There are many problems of pests and diseases in Malaysian agriculture awaiting biotech intervention, such as the papaya dieback disease. Not forgetting our new golden crop of durian also suffer in many disease and pests. For our commodity crop oil palm, this is an example of the that we can target epigenetic modification or karma so that we can get a better fruit. And this technology is possible using targeted demethylation using dCas9 inducer of ROS1 on TET1, and this has been established by Prof. Su Jen Kang lab. We can see that Indonesian researchers have been working on oil palm genome editing. So these are publications, I think, mostly from Indonesia. We haven't seen any Malaysian publications. The same apply for other tropical crops such as the rubber tree, cocoa tree, banana, and also cassava. And there are also potential for other tropical fruit. This is a grant proposal just recently approved in Hawaii on papaya. And then we have the gentian flower, also vegetables. Precision biotechnology will allow ultra-fast breeding, such as the case for this non-sticky elite rice variety transformed into a sticky rice in 2 generations. This was considered a non-GM because there is no trace of transgene. However, there are still challenges to overcome, especially for plant recalcitrant to transformation. Regulatory framework is also a grey area in Malaysia on whether genome editing plant can be considered non-GMO if without any trace of transgenes. And this regulatory framework mainly apply to the edited plant and the regenerated callus. So this is still a hot topic, which I'm not going into details, and we have the whole session on this topic, on whether to regulate genome edited plants. And this map just shows the current situation, and Malaysia still unclear. So I just at the countries such as Argentina, which is supportive of non-GMO regulation, promote local researchers to adopt genome editing and product commercialization. We hope the same will be the case in Malaysia. And I think this conference will serve as a great platform to discuss on this issue. In summary, advanced genome editing tools available for application for precision biotechnology in agriculture for crop and poultry are readily available. Limited genomic resources of local produce will hinder precision targeting. And with challenges of recalcitrant tropical species, we have to leverage on the established tissue culture technology that we in our local researcher for tropical crop improvement. And this adoption of precision biotechnology will promote research and development. We need to increase public awareness of the different modern agri-biotech crop, which is limited at the moment. And local regulatory framework of genome editing need to be made clear. So are we going to have a genome edited durian without the pungent smell in the future? There's something to ponder about. For the last 5 minutes, I would like to share about our research toward precision biotechnology at Imbiosis, University Kebangsaan, Malaysia, which is the first in Southeast Asia and the only Institute of System Biology in Malaysia established 15 years ago. In line with precision biotechnology, we aim to integrate multi-omics with bioinformatics to study plants and microbes for the application in synthetic biology. Some of our research fellows have also embarked on the application of Crispr technology in microbial systems. However, we are yet to perform genome editing in plants. We adopt systems approach, especially transcriptomic, to the study of various tropical species. These include Persicaria minor, Kesum, Mitragyna speciosa, Ketum, Jatropha, Kappa ficus, and Eucuma seaweeds, Acularia agarwood gaharu, and Nepenthes tropical carnivorous plant for molecular discovery. For crop improvement, we work on rice, carica papaya, and Garcinia mangosteen fruit ripenings. We also use the model plant Arabidopsis for functional analysis of genes, our national heritage fireflies, Matissa plana bagworm pest of the oil palm, and the delicacy tiger grouper fish, which is suffering from fibrosis disease. I'm leading the plant functional genomic groups in line with the two research focuses. We perform endosperm culture in an attempt to obtain triploid seedless papaya and also apply transgenic RNA interference approach for the enhancement of flavonoid and carotenoids. We also applied transcriptomic, proteomics, and metabolomic approaches in the discovery of commercially useful enzymes and pharmaceutically valuable bioactive compound from Nepenthes pitcher fluid and pitchers tissues, while answering the fundamental biological question on how hybridization influences the molecular expression and the regulation of protein secretion in the hybrid species of this fascinating family of tropical carnivorous plants. They have huge potential to be exploited as a new bioreactor system for controlled production of plant-made protein in precision biotechnology. Due to high pharmaceutical values of xanthone in mangosteen pericarp, our group also studied in vitro micropropagation of Garcinia species and their recalcitrant seed development and germinations. We performed de novo genome survey, flow cytometry, and cytogenetic analysis, which show that mangosteen is an allotetraploid with a complex genome. This illustrates the challenges of working with non-model tropical species. As you can see in this slide, the common variety of mangosteen on the left and the Mesta variety, the more popular variety on the left with a higher genome size. That's all from me. Thank you all for your attention. I'm happy to answer any questions.